Good morning. Thanks for having me. My name is Sergio Luna. I am a PhD student from Mexico, and here I will present the progress of my research project with the topic Contrasting Effects of Predictor Collinearity and the Transferability of Invasive Species Distribution Models. As an introduction, I would like to start by mentioning that biological invasions represent the second leading cause of biodiversity loss worldwide, and freshwater ecosystems are especially susceptible to their negative impacts. Once established, invasive species are often difficult to eradicate. Considering this, a faster response will usually have greater cost benefits. Therefore, it is essential to identify those species that represent the greatest risk of invasion to justify effective management measures. Currently, there is a large amount of biodiversity and environmental data. The integration of this information associated with the ecological niche concept have resulted in the development of correlating modeling methods to infer species distributions, also known as species distribution models. In the case of invasive species, a paramount consideration is model transferability. Models might have the ability to produce good predictions within the range of values where they are calibrated, but the accuracy of the predictions may decline beyond this domain. Here we can see the relationship between accuracy and transferability across different groups, where aquatic species performed the worst in both metrics. However, it is possible to obtain models with good accuracy and transferability. To ensure the usefulness of models for conservation purposes, it is essential to explore the factors that influence model transferability. In this study, we determine how model transferability is affected by 5 factors known to influence model performance in the context of invasive species using a simulation approach. For this, we use two groups of environmental variables. The first consists of bioclimatic variables from world clim, containing information on precipitation and temperature. The second group are topographic variables from USGS, containing information on the hydrological features of the landscape. Virtual species were simulated using only bioclimatic variables in North America, restricted to freshwater regions, as shown here. For this, 5 variables were selected at random to define the species niche using the two principal components of the environmental space, as is shown here. This niche was then projected across the study area to define the environmental suitability, which was then converted to an occurrence probability using a logistic function. After this, presence points were selected from the environment as a function of the occurrence probability. To define the native range, we assign a dispersal capability at random, with values within this range, which for this species was 143 kilometers. Potential native areas were simulated by selecting presence points within the species dispersal ability, and randomly selecting one as the native area, which is shown here in color reds. The potential invasive ranges were those presents falling outside of the native range, which is shown here in red. From this native range, 100 occurrence records were selected with a probability equal to the occurrence probability times the magnitude of the spatial sampling bias, which was simulated here using the Human Influence Index dataset, standardized with values from 0 to 1. Niche models were built using Maxent from the Dismo package, with the different levels of the 5 factors set out here. The influence of the environmental predictor variables was determined by using two combinations, only bioclimatic variables and both bioclimatic and topographic variables. The effect of the extent of the calibration area was evaluated by sampling the background within a radius of 25 to 1000 kilometers around the presence points. We compared the effect of dealing with predictor collinearity by calibrating models using all variables available or by removing variables with a Pearson correlation higher than 0.7. Spatial filtering of present records is one of the main methods used to reduce the sampling bias present in species data. Occurrences were filtered spatially with a maximum distance from 0 to 20 kilometers. Regularization multiplier is a value that penalizes the use of additional or complex parameters in order to avoid overfit models. The effect of this factor was evaluated by using values from 0.5 to 10. Each model was calibrated using only native occurrences and evaluated according to its predictive capability in native and invasive ranges, the latter to infer model transferability. In both cases, model performance was defined as the Spearman rank correlation of the true occurrences probability and the suitability values predicted from maxim. A total of 960 models were evaluated. Here are the results obtained from the native area. The predictive ability is displayed on the y-axis, and the effect of each factor is shown on a different plot. To summarize these effects, we use linear models. We can see that the greatest effect was because of the reduction of predictor collinearity, which explained 58% of the variability. Unexpectedly, the model performance tends to decrease when predictor collinearity is reduced, and increases when all variables are used. This might be because of predictor variables included in the species niche were removed by the algorithm used, something that should not happen frequently with real species, but it is not straightforward to simulate. The calibration area explains about 14% of the variability, with a pattern that needs to be interpreted with a reduction of collinearity, given that its interaction explains almost the same variability. This will be shown in the next slide. The effect of the other three factors together explain less than 3% of the variability. Here we can appreciate the effect of the interaction between the calibration area, which is displayed on the x-axis, and the reduction of predictor collinearity, shown with these colors. When correlated variables are excluded, performance tends to decrease as the calibration area increases. But when all predictor variables are used, the performance does not vary greatly within this range of calibration areas. Here are the results obtained from the potential invasive range. In general, transferability was poor for this species, given the low correlation obtained. The greatest effects were also associated with predictors' collinearity, interacting with the calibration area extent. Here we can appreciate the effect of this interaction. When correlated variables are excluded, overall performance is higher compared to the use of all predictor variables. This effect was opposite to the one found in the native area, which may result from a higher chance of extrapolation when more predictors are used. The interaction is observed here as a slight increase in performance with the extent of the calibration area here, and a slight decrease in performance with the extent of the calibration area here. In conclusion, it is possible to obtain models with high functional accuracy within the native range of the species, but the transferability might be reduced in areas with invasive risk. Excluding correlated predictor variables and the extent of the calibration area were the main factors affecting model performance, with some contrasting effects on the first factor. It is possible to increase transferability by reducing the number of predictor variables and selecting broad calibration areas. Further analysis of the relationship between discrimination accuracy and metrics such as ROC and transferability is needed. Thanks for listening.